Gene transferring bacteria, and uh, today we would like to start with conjugation, for which cell to cell contact is required. So the conjugation, the discovery of conjugation dates back to 1946, when Joshua Lederberg and Tatum, they performed an experiment with two bacterial strains. So I think conjugation you people have studied. So would you like to tell me what all you have so that we can have a quick revision and move further? Class?
Yes, ma'am.
Yes.
Like in the case of conjugation, there is one cell which have a specific plasmid known as F plasmid, and this and the cell which have F plasmid, it is able to form something called a pili, and that pili is the way by which a cell can transfer a DNA into another cell. So when a cell has F plasmid, it can form a pili and transfer DNA to transfer plasmid to another cell. The cell which don't have the plasmid. When there is this plasmid in one cell, and it came into contact with another cell which doesn't have, so it forms a pili, and then the transfer happens through that. And
Okay, and do you
okay,
remember the experiment by Bernard Davis, the famous YouTube experiment?
yes, ma'am.
Ma'am, I guess I can explain it.
Ha ha, Ashi, tell me.
Ma'am, so the previous experiment, the first one that you showed, in
Mm-hmm.
that we use auxotrophs,
Mm-hmm.
and we observe that when we like auxotrophs are basically are basically deficient, as in they cannot produce particular protein per se.
Right.
So
Yes.
when they cannot produce particular amino acid, so what happens is when we mix two auxotrophs, we observe that the colonies are no more deficient of either of the proteins
Hmm.
because conjugation takes place.
Okay.
But when individually we observe two different auxotrophs, we see that the growth would be that the particular amino acid expression would be missing in them in the two
Correct.
different colonies.
Correct. Very good. And then this one.
I guess here they placed a filter and they discovered that for conjugation to take place, it is important to be in contact with each other.
Yes, this very good. Okay. Then what else you have studied? I think you have studied this type of conjugation from F plus to F minus.
Yes, ma'am. We studied for the fertility plasmid and we studied HFR also. That
Okay,
when
so
it is
let's go over this.
integrated inside the chromosome.
So let's go over one by one. So maybe starting with this one.
Right, ma'am. Yes, ma'am. So, when the fertility plasmid is present, the strain, the bacteria would be, it would be a F plus cell, and when it is absent, it would be a F minus cell. Now,
Yeah.
two F plus cells would never conjugate.
Mm-hmm.
Whenever conjugation takes place, it would occur from F plus to F minus, and the plasmid would replicate, and a copy would be sent to the F negative cell as well, and then
Yes.
both the cells would become F positive
Okay.
after the replication procedure completes.
Okay. Very good. Now this one. HFR.
So what happens is at times the F plasmid combines with the chromosome itself, and then we call the cell HFR cell. I guess high frequency of recombination is
Yes.
something that they're.
Yes.
When the HFR cell forms a pili a structure or conjugates with an F minus cell, sometimes a part of the chromosomal DNA is also transferred to the F minus cell. So
Right.
the chromosomal DNA along with the along with some part of the along with some part of the F plasmid gets transferred to the F minus cell, and now the new F minus cell, that is homologous recombination that would be occurring here, would have two copies of the chromosomal part and some part of the F plasmid.
Yes. Okay. And then the third one, the F prime.
Right. Yeah, so what happens is sometimes instead of the chromosomal DNA directly being transferred, the chromosomal DNA, some part of the chromosomal DNA is present in the form of plasmid, as in with the F plasmid, we have some amount of chromosomal DNA also. So when conjugation occurs with the F minus cell, some part of the chromosomal DNA and the F plasmid gets passed. So when the conjugation will take place, the F minus cell would have the F plasmid and some part of the chromosomal DNA present as another plasmid known as F prime.
Okay. Okay. Anything else that you have studied?
Ma'am, I guess we studied these types only, like as far as I remember.
Okay, okay. Ratnesh, would you like to add something?
No, ma'am. I have not remembering all of these. Like I didn't watch all the videos of microbiology. I just read a few articles and from book. And in that the book which I read, all the things was not there right now.
Don't worry. So then I think Ashi has covered more or less everything, but then maybe in the F prime, what is F prime and how is it created? So for that it is important to see the map of the F plasmid and why this F plasmid is conjugative. So for conjugation to happen, right? So for conjugation to happen, a set of proteins are required, and these proteins are called proteins for transfer because they help in the transfer of the plasmid, and these genes are actually called as TRA genes. Genes, and it's a large number of TRA genes like TRA A, TRA I, TRA Q, TRA B, TRA C. Many TRA genes are involved, and in fact if you see in the map of the F plasmid, so this is the map of the typical F plasmid, and you can see that this is the transfer region. So you see almost 1/4, more than 1/4, almost I would say 1/3 of the plasmid part is actually containing the genes for transfer region. So this usually encompasses about 33 kb of the total plasmid, right? So 100 kb is the size of F plasmid. 100 kb is the size of the F plasmid, and the TRA genes, they encompass about 33 kb. So that means there are a number of TRA genes involved in this. The other important feature of the F plasmid is, if you see carefully, it has an ORI-T. Over here, it has an ORI-T. This ORI-T is called as origin of transfer. Okay? ORI-T. T is for transfer. So, for plasmids to be conjugative, other than that, these rep proteins are for replication, TN is the transposon, IS elements are the insertion sequences. IS elements are insertion sequences with the help of which this plasmid can integrate with the chromosome. So, IS elements can combine with IS elements in the chromosome. Alright. So, can you people hear me or no?
Yes, ma'am, we can hear you.
Okay, okay. So on the basis of this, actually plasmids can be classified as conjugative plasmids, or second category is mobilizable. Conjugative plasmids are the plasmids which carry all the TRA-genes, right? All the TRA-genes plus ORI-T, can be self-transmissible. They are self-transmissible. On the other hand, mobilizable plasmids are the ones which carry ORI-T and which carry MOB protein. MOB is for mobilization, and which do not contain any of the TRA genes. So in such cases, the TRA genes, the TRA proteins are actually provided by the host, by the chromosome, by the host strain. So mobilizable plasmids can be small in size because all the TRA genes will be encoded by the host chromosome, but origin of transfer is must in both of them. It is because of this origin of transfer, the actually helicase binds over here, and then the MOB protein and the helicase binds at origin of transfer, and then single strand is transferred from 5' to 3' direction in the recipient strain. So for conjugation, actually what we require is, let's see what are the requirements of conjugation. Conjugation, we require a donor strain, and as we discussed, cell-to-cell contact is required for conjugation to happen. So now the way it is done is, class, again you start with single colony, right? Grow the cells to mid-log phase. So have you studied this, like how exactly to perform in the lab? Should I skip this part?
Ma'am, we haven't studied it.
No, ma'am, we have not studied how to perform that in lab. We have just studied the theoretical parts. And
okay, so you single colony, you take, grow the cells to mid-log phase, again because the cells are very healthy, and then so both donor and recipient, they are grown in separate tubes. Now what you do is, you need to perform what is called as mating. So for this mating to happen, you can actually perform mating on solid medium or on liquid medium. So the way it is done is, on solid medium, the way it is done is, you take a Petri plate and then place a filter membrane containing donor and recipient. Okay, so the way it is done is, you can take actually 0.2 micron filter paper, pass the grown cells of, first it is important to spot the recipient. So first you pass the recipient, collect the cells on the filter, then again on the same filter membrane you pass the donor strain. So at the bottom you have recipient, at the top you have the donor strain, and then you again allow the culture to pass through the filter. So on the filter actually you will have cells, both donor and recipient. Okay, and then what you do is, you carefully place this filter with the help of a sharp surgical forceps onto the Petri plate, and then leave it overnight in the 37 degrees incubator. Next morning what you do is, you swap the cells from the filter, and then this is important. So then the term that is used is, these are called as transconjugants. Transconjugants are selected on antibiotic plates or selective media plates, which are selective for both donor and recipient. What I mean is, class, that for example, if the donor strain is kanamycin resistant, and let's say the recipient has, for example, streptomycin resistance. Then in the trans conjugate, you need to select for double antibiotic plate, because if you select for kanamycin, only donor will grow. If you select for recipient, that is streptomycin, only recipient will grow. So trans conjugates are the ones which will grow on both. So that means you will have to see that the kanamycin is now transferred to the streptomycin, right? So ultimately you can select it on double antibiotic plates. So I think this will become clearer with the help of an example. So I'll give you an example of performing integrations. Transfer by. So for E. coli, the strain that is used is usually SM10. And this contains all the tragenes. This is the strain E. coli SM10, which contains all the tragenes. So this is usually the donor strain. And in this you can actually transform the plasmid. So now the class knows how to do the transformation. So you can put the plasmid, for example, which contains the GFP acid. Now let us say, so the important feature of conjugation is that conjugation can be done from gram-negative bacteria to another gram-negative bacteria. Conjugation can be done from gram-negative bacteria to gram-positive bacteria, and conjugation can be done from gram-negative bacteria to even Saccharomyces yeast also. So that means conjugation can be done across boundaries. The other thing before I go into the detail is that yes, like Ashi mentioned, that for conjugation you require a pilus, and this pilus is made up of a protein which is called pilin, which is actually encoded by the gene called TRA, and the protein is actually called TRA, which actually ultimately exists as propylene and then converted into pilin, and this actually polymerizes to form the pilus, to form the tube, the pilus. The way the process works is, I have a figure to show you. So the way the process works is, the pilus binds, and then there is something which is called retraction of the pilus. So you have a long pilus, but then this is a protein, so this depolymerizes and brings the other cell in close proximity. Thereby the contact between the donor and the recipient is stabilized, and ultimately the transfer takes place, and finally as a result of depolymerization the two are ultimately separated out. So this is how the conjugation process actually takes place, when the pilus, long pilus is formed, but then this pilus has a tendency to polymerize and depolymerize. As a result of this depolymerization, the retraction takes place, as a result of which the donor and recipient come in close proximity, and the contact is stabilized, and then the transfer takes place. But when we talk about gram positive bacteria, so gram positive bacteria, you do not have pilus. So then how exactly conjugation takes place in case of gram positive bacteria? So in case of gram positive bacteria, there are certain proteins which are called as adhesins, present on the surface of the bacteria, and these adhesins actually help in bringing the two cells in close proximity, and as a result of which conjugation takes place. So conjugation class is actually a very slow process. So the transfer of the DNA by conjugation is a very slow process, and that is why people realize that we can use this process of conjugation to map the chromosome. So let's see, we were here.
Ma'am,
Yes, tell me.
I read that it is a very fast process, that like it takes just 5 minutes to conjugate and transfer the gene.
Okay, so yes, but if you talk about transformation, it takes 1.5 minutes, right? So in that sense, it is a slow process, and I'll tell you why this slow process is preferred. So, and why it can be used for integration of genes. So I think this is more or less what Ashi has explained, that F plus can become F minus. So here I would like to add a little more detail onto it. So when we talk about HFR cell, so HFR cell is high frequency recombination cell. So in this case, the F plasmid can combined with the chromosome, and this recombination is facilitated with the help of what is called as IS elements. So the point is, if you delete IS elements, you will not have an HFR cell. So it is the IS elements which actually aid in the recombination, and that is why you created HFR cell. The other point about the F prime plasmid is that the HFR cell which is shown in this figure. So in this figure, the black part is the chromosome and the gray part is the F plasmid. The plasmids which can exist as independent entity as plasmid and which can also integrate are also called as episomes. So episomes are the plasmids which can integrate in the chromosome. Okay, so that means F plasmid can exist as an episome as well as a plasmid. So what happens is that this plasmid, this F plasmid, which is integrated in the form of an episome, it can actually loop out, and as a result of this plasmid is created, which is called F prime, which is different from F plasmid in the sense that when it is popped out from the chromosome, it also brings along some part of the chromosome. So this is the only point that I wanted to add over here, Rashi. So F prime is actually, the way F prime is created is because of the feature of this F plasmid which can exist as episome. Okay, so now the strain HFR can be used for mapping the chromosome. So when we talk about mapping of the chromosome, the E. coli chromosome, when sequencing was not done, right? All the time we discuss that restriction mapping is done for plasmids. So how do we map the genomes? We all the time we used to discuss this, that we cannot develop a restriction map of the chromosome. So can we map the genome without sequencing? So people in 1970s or 1980s, they came up with the idea that using conjugation followed by interrupted mating, map the genome. And the way it is done is, and that is how people realized that, and people prepared the 100-minute map for E. coli chromosome. Minute map for E. coli chromosome is, so E. coli chromosome is how big? 4.6 megabases. You remember that? So for transfer of this 4.6 megabase pairs, it takes 100 minutes. So then, how people realized that this 100 minutes is taken for this transfer, and how we can prepare the map of the E. coli chromosome based upon the interrupted mating and what is interrupted mating. Have you studied this? Would you like to tell me?
Ma'am, no, I have not. Ma'am, I don't know.
Ashi, have you studied this?
No, ma'am,
Okay.
not yet.
Okay. So let me explain you this, that what is interrupted mating first. There is a very nice video which I will be showing you in a second. So in interrupted mating, as the name itself says, mating That means both donor and recipient are mixed in liquid or in solid medium, right? And then what you do is you interrupt the mating for 5 minutes. So this mating is interrupted by mixing with you can just put it in a vortexer or mixer blender. So for example, you have two test tubes, one containing donor, one containing recipient. Mix. Put it in a blender. So the moment you put it in a blender or you vortex it, the pilus, the contact between the donor and recipient will be broken and the mating will be interrupted. So the donor and recipient will get separated and mating will be stopped. So that is how what people did was after every 5 minutes you take an aliquot. So you have two test tubes, one containing donor, one containing recipient. You take an aliquot of donor, take an aliquot of the recipient, mix it carefully, leave it aside for maybe let's say 5 to 10 minutes, and then you start doing the sampling. So after 10 minutes, for example, you take an aliquot, put it in a vortexer or blender, and then separate it. After another next 5 minutes, again take an aliquot, put it in a vortexer or blender, separate it. So this you keep doing for about 100 minutes, and then you score for transconjugates. Okay, so now what will happen in the transconjugates? So in the transconjugates, as Ashi had mentioned, that you will actually score for the transfer of the, for example, amino acid biosynthesis genes, or for example some visible markers whose transfer you are going to score for. Okay, so let me show you how exactly, maybe from one figure. So this is a typical figure of interrupted mating. You can have markers, for example, ASI is azithromycin, LAG is, for example, you can have GAL for galactose, you can have LAC for lactose, you can have different markers, and you interrupt the mating and then you score for the number of these positive colonies. You're getting my point or no? And then maybe I'll come back to this in a second, the mechanism of transfer. But before that, let's see from this figure how exactly the transfer takes place. So you have, for example, this is the strain which is HFR, streptomycin sensitive, and these are the markers: threonine, cysteine, and phenylalanine. Okay, amino acid biosynthesis genes. This is F minus strain and this is HFR strain. The F minus strain is the recipient strain. The recipient strain is streptomycin resistant. The donor strain is HFR strain and it is strep sensitive, right? And what you do is you mate this donor with the recipient. Okay, and these are the mutants, and these, the plus ones, are the ones which are positive for these amino acid biosynthesis genes, and A13, G22, A10 are the mutants. That means at the 13th position there is a mutation, at the 22nd position there is a mutation, at the 10th position there is a mutation. So the point is, as a result of this mutation, the biosynthesis, the amino acid biosynthesis genes are not functional. Okay, now
Yes, ma'am.
now what will happen is the conjugation, you will allow the conjugation to take place, right? So so ultimately, as a result of mating, right, so what can happen? Any guesses? So from HFR we are allowing the mating from HFR to F minus.
Ma'am, recombination will happen and
Yes, and
transfer of genes can be transferred and means recombination just or what
yes,
can
but right, but then what will be the direction of the transfer? Point number one, and depending upon the so let me explain you. So when we have the blue arrow that is shown over here, this blue arrow is showing the origin of transfer. Okay. So that means origin of transfer is the place from where the transfer will take place. Okay, so the markers will start moving, and ultimately you see this figure. So the order of transfer will be phenylalanine, cysteine and threonine, right? So what you see is that 15 minutes phenylalanine was transferred, in 25 minutes cysteine was transferred, in 35 minutes threonine was transferred. So in order to explain you this figure and this process, you need to understand that phenylalanine, the number of cells are 75% cells had phenylalanine, because this was the first marker which was transferred. Are you following this or no?
yes ma'am.
And then the second marker was cysteine and the third marker was threonine. So this is the order of transfer into F minus, right? So likewise class. So here for example we have three markers, but likewise in the entire E. coli chromosome you can have different markers, and you can select a donor strain. You can have different donor strains. So for example this is HFR-A, but then you can have HFR-B where for example the arrow is here. You can have HFR-C where the arrow is for example here. You can have HFR-D. So all I'm trying to say is you can have different types of HFR strains, and these HFR strains, they are different with respect to the site of integration of the F plasmid in the E. coli chromosome. Following or no? Is this making sense or no?
Yes,
Ma'am,
ma'am, it is.
can you please repeat ma'am? Yeah, once.
Right. So what I wanted to tell you was the way I'm showing you this figure where the blue arrow is the origin of transfer, right? And HFR is the strain where the F plasmid has integrated in the chromosome, right? Over here.
Yes ma'am.
Now the point I'm trying to tell you is that in the E. coli chromosome, there are multiple places. It is not that F plasmid can be combined at only and only one place. There are multiple places in the E. coli chromosome where the F plasmid can integrate with the help of IS elements.
Yes ma'am, yes, that I understand.
So as a result of this, different types of HFR strains can be created,
Yes.
where you have F plasmid integrated at different locations. Yes.
Yes ma'am, yes, that I understand.
Right. So as a result of which, when conjugation will take place from HFR to F minus cell, right? And if you try doing it for multiple such strains, you will be able to generate a map of the chromosome based upon the transfer of markers.
Yes ma'am, based on the percentage, I can say that what is the arrangement of this.
Exactly. based upon the number of colonies. So for example, on the trans conjugates, you get 75% colonies which are phenylalanine positive. Let's say 20% colonies are threonine positive, 0% colonies are for example azithromycin positive, something like this. Likewise, you do conjugation where for example the F plasmid is integrated here, it's another HFR. Again you perform conjugation and again you score for the positioning of the markers, right? So this you can do with multiple HFR strains, and you can actually so you can do this for up to 100 minutes or 120 minutes or even more, and then try to develop a map.
Yes.
Yes, followed.
Yes. Yes, ma'am. This much I understand.
And this is a typical diagram to show you that how exactly this recombination takes place. So this is origin of transfer, and this is the F plasmid. Here it's the bacterial chromosome, which is shown as A, B and C are the markers. And for example, the recombination takes place here. So origin of transfer is here. So you see that the way it is integrated is you have E and then followed by D in another figure. So this is important, and then I'll show you a typical problem which you can try solving it. So over here again it is shown that the transfer of the plasmid takes place from an HFR cell to the recipient cell, and what you can see is that the markers, for example genes D and A, are transferred early, C, B and E are transferred late. So accordingly you can prepare a map. So the example that I wanted to show you is this one, right? So this is the example which you can try solving it. So this is where you have 4 HFR strains derived from the same F plus plasmid. So that means, as I told you, this F plasmid can integrate at multiple places. So you have strain 1, strain 2, strain 3 and strain 4, and now you need to determine the order of markers in the chromosome. So just note it down and then try solving it. And then I'll show you a quick video. Have you noted it down? Should I?
Yes, ma'am. We don't have to solve now. We can solve.
Yes, you can solve it later, and then if you have issues, we can discuss this on Friday's class.
Yes, ma'am. So I have taken the screenshot and I can.
So then maybe I'll show you a quick video just to show you the interrupted mating part, and then we'll start with transduction.
Interrupted mating and recombination experiments
Can you
to
hear
map
us,
genes in bacteria. And this video is
Yes,
particularly
ma'am. We can hear.
students enrolled in my BIS 101 course or other BIS 101 sections.
Just move it further.
At UC Davis. So we're going to briefly go over conjugation, specifically focusing on conjugation between an HFR cell and an F- cell. And in this case, in the HFR cell, the fertility factor plasmid has been integrated into the bacterial chromosome, where the bacterial chromosome is indicated by teal and the fertility factor is the red region. And you can see that in the F- cell.
So I hope you will now, so F plasmid is the one which has, it's called fertility plasmid because it can provide the genes for F pilus, but then F is not the only conjugative plasmid. There are many plasmids, for example, even R plasmid is conjugative. So there are many other conjugative plasmids as well.
The bacterial
Yes, ma'am. This was a confusion I had
have the
already.
fertility factor integrated in it. More so, the F- cell does not have the fertility factor. And as we know, the fertility factor encodes genes that are necessary for building the pilus, which allows for conjugation. Now we want to focus in briefly on the black and the green region indicated in the bacterial chromosomes. For in the F- cell, it's the black region, and in the HFR cell, there's this green region. And these two regions indicate areas of homology.
So the another terminology that I didn't mention is, so the recipient strain and the donor strain. So there is a time when they both have the same copy. So for example, the donor strain has the homology region. So this is called as endogenode, and the recipient and the donor strain which brings the homology region, it is called exogenode. And when the two are together, it is called as merozygote. So this is just the terminology.
Meaning that they're regions of the chromosome that encode for the same DNA sequence. So now let's examine what happens during conjugation. During conjugation, a pilus forms from the HFR cell that builds a physical contact with the F- cell. And then, starting at the fertility factor origin, the bacterial chromosome is replicated through a process called rolling circle replication, where replication begins at the origin, indicated by this arrow, and continues along the bacterial chromosome. And as replication is occurring, a single-stranded DNA copy is then transferred through the pilus to the F- recipient cell. Then, once in the F- recipient cell, that single-stranded DNA copy is replicated to form a double-stranded DNA molecule. And the result then is that part of the fertility factor, because the origin is in the fertility factor, and then the associated bacterial chromosome next to the origin is transferred into the F- cell. And if we remember then the regions of homology between the two bacterial chromosomes, one in the F- cell and one in the HFR cell, they share regions of homology, which promotes and allows for crossing over or exchange between the complete genome of the F- cell, called the endogenote, and the partial genome that was transferred over through conjugation, called the exogenote. And so at this current stage, the cell has two copies of one segment of the DNA. One copy is part of the endogenote and the other copy is part of the exogenote, resulting in what's called a partial diploid or meriploid. And if such a crossover event happened, where it's a double crossover or quadruple crossover, it results in exchange of genetic information between the two bacterial cells, where now the F- cell has integrated in the bacterial chromosome the DNA sequence from the HFR cell. And the remaining segments of chromosome will be lost in subsequent cell growth and division that were not integrated into the bacterial chromosome. So that is a very brief overview of bacterial conjugation, which is only one mechanism for how bacteria are able to exchange and take up genetic information. They can also exchange and take up DNA through transduction and through transformation, but those are not the focus of this video. However, we can use then this process of bacterial conjugation to map genes on the bacterial chromosome through interrupted mating experiments and recombination mapping experiments. Let's first walk through an example of an interrupted mating experiment. And in this experiment, we are mating an HFR cell that is prototropic for Lu, Trp, and Arg and sensitive to the antibiotic kanamycin. And we're conjugating this with an F- cell that is oxytrophic for Lu, Trp, and Arg, but resistant to the antibiotic kanamycin. So this F- cell needs to have Lu, Trp, and Arg added to the media for it to be able to grow. So we take the HFR cells and the F- cells and we mix them together in the media. So now we have our tube that contains both the HFR and the F- cells in the same physical space, allowing for conjugation to happen. And then what we do at different time points, we're going to take samples from this conjugation mixture and plate it out onto different media conditions. And the first thing we want to do in our interrupted mating experiment when we plate these cells out is to select for F- cells, because these are the ones that are going to be receiving DNA through conjugation. And it is the time in which the DNA segments from the HFR enter the F- cells that informs how we are able to order the genes relative to the fertility factor origin. So in order to do that, when we take our time point samples from this conjugation mixture, we need to select for the F- cells only and therefore get rid of the HFR cells. And we do that by adding kanamycin to the minimal media. And if we remember, the HFR cells are sensitive to kanamycin. So adding kanamycin to the minimal media on the different plates that we plate these out on is going to specifically kill the HFR cells, allowing us to select for the F- cells after conjugation. Now, when we take our time samples and plate them out, we're going to plate them out on different media conditions. All of the conditions are going to be minimal media plus kanamycin. But then we're going to have different conditions where we are able to select for whether the F- cell has received the prototrophic marker from the HFR. So in the first plate, we are adding leucine and tryptophan to the media. So that means that the F- cells we are selecting for are the ones that have then received the Arg prototrophic marker, because the cells that grow in this condition are able to grow in the absence of Arg, which the F- cells alone cannot. So the only cells that are going to be able to grow in the absence of Arg are those cells that have received the prototrophic marker from the HFR chromosome that was copied and transferred over during conjugation. For the second condition, we are selecting for F- cells that have received the TRP prototrophic marker from the HFR, because we are not putting tryptophan in the media. So only the F- cells that have received that prototrophic marker through conjugation will be able to grow. And then in this third condition, we are selecting for F- cells that have received the prototrophic Lu marker from the HFR, because these cells would be able to grow in the absence of Lu. So again, we mix our cells and we allow them to undergo conjugation. And at different points after mixing the cells, we then take samples from this master mixture and plate it onto different media conditions. So if we do that, if at time 0 we take a sample from our master mixture and plate it out under these different conditions, we would expect no colonies to grow, because there hasn't been any time for conjugation or transfer of bacterial chromosome DNA from the HFR to the F-. Let's say we let some time go by and then take another sample. We take another sample and plate it out on these different conditions. So after 10 minutes, we now see that we have some colonies starting to grow in the absence of tryptophan, or TRP. And what this tells us then is that the prototrophic TRP gene was the first to transfer over during conjugation, meaning that it's the closest to the fertility factor origin on the HFR bacterial chromosome, so O for origin. So then if we wanted to map these genes relative to the fertility factor origin, we would say that TRP is the first one, or the closest. So now we're going to let some more time go by, 10 minutes later. So now after 20 minutes from the start of mixing these cells or allowing them to conjugate, we take another sample and plate it out under the different media conditions. And what we see now is that after 20 minutes, we see that cells or colonies are surviving in media that is lacking arginine. So what that tells us then is that the prototropic arginine marker was the second marker to transfer over to conjugation. And so now we can order arginine as the second closest marker to the F factor origin. Now if we let a little bit more time go by, and 30 minutes after mixing these cells, we take another sample and plate it out under these different conditions, we now see that we have cells able to grow in the absence of leucine. That tells us then that of these three, leucine was the last prototropic marker to transfer over during conjugation. So leucine is the furthest from the F factor origin. So by analyzing this interrupted mating data, we were able to deduce the order of these three markers in relation to the fertility factor origin in the bacterial chromosome of the HFR strain. So now that we've ordered our genes,
So hope it was clear, students.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay. So maybe let's start with, we have 5 minutes. So maybe quickly let's start with transduction. So tell me what all you have studied about transduction and then we'll go from there. Who's going to try? What is transduction? Have you studied transduction, generalized and specialized? Have you studied?
No, ma'am. We have just
Yes,
studied transduction.
Oh, ma'am,
Ma'am,
perhaps we have
both are
studied both.
Okay, so then tell me.
Ma'am, sometimes when a virus infects a bacteria and there's some sort of a genetic switch which decides whether it will start producing its own proteins to produce more bacteria, or whether it will remain dormant in the bacteria's genome for several generations. And when that happens, and when another bacteria comes and infects this bacteria which already has a viral genome, and it starts producing more viruses, it also copies some part of the other virus's genome and packs it inside, and then transfers it to another bacteria through the same cycle.
And
That's all. So yeah, there's the lysis-lysogeny cycle, and that's controlled by some sort of a switch which decides.
okay. So when we talk about bacteriophages, they are classified into multiple types based upon the structure. So for example, you can have, based upon the structure, we can have filamentous phages, and it is the filamentous phages which are used in the earlier days for sequencing purposes. So filamentous phages, the examples are F1, D. So all of these are filamentous phages. They are really filamentous in shape. That means they are 6 nanometer in diameter and 11 nanometer in length, and the genome is roughly I think 6.7 kb, so it's really small. The other phages that you will be studying are lambda phage and lambda phage, it has an icosahedral head and tail, and the genome size is approximately 50 kb. So lambda phage is the phage which is actually called a temperate phage. So when we classify the phages are classified as virulent phages. Virulent phages are the ones which can exist, which can lyse the cell, which show only lytic cycle. So whenever they infect, they actually lyse the cell, and these are therefore called as virulent phages. Temperate phages are the phages which can exist as both lytic cycle and also show lysogenic cycle. So lysogeny is the stage when the phage DNA integrates in the chromosome, and lytic cycle is when the phage actually packages, the phage DNA is packaged and the cell is lysed. What about filamentous phages, the M13 phages? So these phages, they are actually virulent phages, but they are unique in the sense that they do not lyse the cell. So they do not integrate in the chromosome class, but they do not lyse the cell either. So the way they work is because they are so because of their filamentous shape, and because they are so thin in diameter and so small in length, that they just escape from the pores of the cell membrane. So whenever filamentous phages they infect a bacterial cell, they do not show lytic cycle, they do not show lysogenic cycle. Instead, what you will see is that filamentous phages, approximately 1000 phages are produced in an hour. So it is not that it is a slow process. Approximately 1000 phages are produced in an hour, but they are released lysing the cell. What you see is that you see the regions of reduced growth in case of filamentous phages, because the cell's growth will be reduced, but then they will not be lysed. On the other hand, when we talk about lambda phage, when they show a lytic cycle, you will see what is called as plaques, clearance zones, and it is these plaques which contain phages, and lysogenic cycle when it is integrated in the chromosome. So when we talk about transduction, so which is transfer of DNA from the virus to the bacteria, so which phages are used? Any idea? Have you studied this?
No, we don't know about the
Ma'am, can you
phages.
please
Use the
repeat the
lambda
question?
phage.
Huh?
Can you please repeat the question?
So I said ki for transfer of genetic material, which phages are used? Which phages are preferred?
Lambda phages.
Okay.
This we haven't studied, but yeah.
This you haven't studied. Okay. So, so for the phages that are used are actually P1 phage and lambda phage. Both are used for transduction. So P1 phage mediated transduction is called generalized transduction class. And why it is called generalized transduction, I'll be telling you in a, or do I have time? And lambda phage transduction is called as specialized transduction. So we are going to discuss the differences between generalized transduction and specialized transduction, and how they can be used for manipulating the genome, how they can be used for engineering the genome and for transfer of genes in the next class maybe. So I'll stop here and